Hello, good morning. Thank you very much for the invitation and for allowing me to present Hallura today. Hallura brings the breakthrough cross-linking technology of hyaluronic acid, which offers the most natural HA filler and with unprecedented tolerance and best-in-class efficacy and duration. So the core of the technology is what we call the BioLink matrix, which is a lock-key cross-linking between long polymer chains of HA. So the current products are typically cross-linked with BDD, which is a random reaction and with quite low efficacy. So the main thing happening during BDD cross-linking is a shortening of the polymer of HA into smaller chains, which make HA losing a lot of its intrinsic elastic properties. And right after cross-linking, the gel must be grained into particles in order to wash out all the toxic residues. So you end up with a material which is made of granules, losing a lot of intrinsic elasticity, and that needs to be hard in order to lift the skin. What we have used is a cross-lock-key approach between long chains of HA. This lock-key or click chemistry approach has been recently awarded a Nobel Prize and had many medical applications for cancer drug release, tissue engineering, or diagnosis. We are using it for the first time into aesthetics. The purpose is to keep the chains of HA as preserved as possible and treated in very smooth condition during all the manufacturing in order to achieve very high elasticity and very high homogeneity of gels. So we performed a lot of the typical rheological experiments, but this video can tell the story in a few seconds only. So you can see the elasticity of Hallura gels, which then the elastic strength is tuned by the concentration of HA. With a lower concentration for lips, for instance, we have elastic gel with lower strength, and for volume, a higher concentration for higher strength. This is one of the core aspects of the technology. If you consider the orange curve, which is the raw material, the molecular weight distribution of hyaluronic acid as a raw material, it's about here 1.6 million Dalton. When this HA is treated during BDD cross-linking, the molecular weight shifts dramatically to the left, and it's about only 0.2 million Dalton. It's 8 times less because of the shortening of the chain. With Hallura technology, we are able to maintain inside the cross-link gel very high molecular weight of HA, which allows to keep the natural elastic properties of the polymer, and also other biological properties of such as anti-inflammatory effects. So the application of this technology leads to completely different formulation with very low concentration of HA, so up to 4 times less HA than in a current current product, sorry. We avoid using any toxic material. The HA is more natural, it's less cross-linked, it's less chemically modified, there are less impurities, less soluble HA, and lower hardness for higher elasticity. So we have run many preclinical models showing in animal models or ex vivo human skin, very good integration of the product in the skin. And thanks to a lower concentration of HA, lower density of HA, it favors the natural ingrowth of tissue inside the skin. So we have completed our first clinical study in Europe. There were 3 arms for different indications. In each indication, different formulations were studied. And the purpose was to evaluate the safety for 12 months, sorry, and also to assess the effectiveness versus pre-treatment. So global aesthetic improvement was very high over 12 months in the view of the patient, view of the physicians. This is for lip injections. Also for NLF corrections, satisfaction was very high. Also for cheeks enhancement, a very high satisfaction, over 80% over 12 months. So although the study, it was an open label study, it was not comparative, we can illustrate the effectiveness of the product by comparing the response rate we obtain at 12 months versus what is published in the literature for comparative product using the similar grading scales. So it illustrates the high effectiveness at 12 months on the 3 indications. Safety was very good during the clinical study. Adverse event, there were no delayed adverse event. And in terms of the tolerance score, they were also very positive. In yellow, you can see here the rate of patient reporting swelling 8 days after treatments compared to the typically observed reporting of swelling for other products. So it's much less. Also in other category, less pain, less firmness, less lumps. So overall, a very good satisfaction with the treatment. Few before and after pictures. This is after a lips enhancement. It's 12 months after treatment. So you can see very natural and very long-lasting result. The testimonials of patient and doctors enhance the fact that the integration in the skin is very good. Predictable and forgiving, meaning less post-treatment reaction, and a very natural sensation in the skin. So this is after nasolabial fold correction, and this is a cheeks enhancement. Very few patients were followed up to 18 months, only few of them, but you can see that even after 18 months, results on the lips are very natural and very long-lasting. So in summary, we address the 3 main facial indications. All products contain lidocaine. They have increasing lifting force, and we also seen in ex vivo skin model that we have an improvement in elastin and collagen around the implant. So I think we are preparing a next generation of fillers which will have which will be significantly better than the current product and can enhance the general injection experience for the patient during treatment, in the post-treatment reaction, immediate and also long-lasting. And the product is the most natural in HA because it's less concentrated and less modified, well adapted for the skin, integration into the skin, and providing extreme natural sensation after treatment, which is related to the new mechanical properties with high elasticity and avoiding hardness. Thank you very much for your attention.
Thank you. Very exciting new product. Thank you very much. What IP strategy are you thinking about when you think about just the world of HA fillers? What are you going to do to differentiate yourself from an IP perspective?
Yeah, so the first patent we submitted is really covering the polymer of HA cross-linked with our material. So this was this lock key cross-linker. So the first patent protect really the polymer itself, so the equivalent to the API, and we have subsequent patent for specific formulations, addition of non-cross-linked HA or very specific formulation with extremely low extractables.
Oh, that's interesting. Thank you.
Hey, Stephane. I've worked with dozens, maybe over 100 people that worked in hyaluronic acid formulation, and acknowledge you as one of the most respected scientists in HA, and I respect you immensely. And so I believe things you say, but you're saying here the BioLink matrix, and you're saying that's the most natural. How do I know it's not just another chemical linker here, cross-linker, and it's just different than BDDE?
No, so there is no magic. So there is a cross-linker, that's for sure. The most natural, we claim it's most natural based on the fact that the concentration of HA is lower in the product, the modification of HA is lower. So if you do the simple maths, the amount of non-HA material in the product is the lowest. So it is the closest as natural HA, but of course we are using 2 molecules that are safe, non-toxic, that are pre-installed on hyaluronic acid, and that are able to connect in this click chemistry with this click chemistry approach.
So there is a chemical cross-linker there.
Yeah, there is a covalent cross-linker, which is the only way to make the product last for very long in the dermis. So there is a chemical cross-linker, a covalent cross-linker, but we aim to use a completely different approach with being convinced that HA can provide much better material using this chemistry approach as compared to the typical approach.
All right. Well, maybe Irina have a second question, but maybe you want.
Well, just to follow up on that, you're showing very long-lasting effects with some of these molecules. Have you tested the ability to correct with hyaluronidase? Because as we know, that is a critical element for these products.
Yes, sure. We checked that, and our products are completely dissolved with hyaluronidase. So we haven't checked in vivo, of course, but in all tests, laboratory tests. And so the cross-linking does not inhibit at all the hyaluronidase action. And because it's less concentrated in HA, it's less dense, it's very prone to degradation by hyaluronidase.
And what has been the feedback from the physicians doing injections in terms of the force necessary for the actual injection? Is there any, because you're showing a much more viscous material?
Yeah. So that's it's a very good question. So the use of the product is very easy. The extrusion force is equivalent to a high concentrated product. So because the gel is completely homogeneous, even though it's 4 times less concentrated, you need to apply the typical force that you apply with a higher concentrated product. So the use was described as quite easy. It's easy to position, easy to mold, like the elasticity doesn't prevent the modeling. And so the feedbacks were very good. And no, you don't need any specific training in order to be able to inject.
Let me ask another question. It is, we say it's cross-linked with BDD, it's cross-linked with BDD. We don't, and you brand it as BioLink Matrix. So my question maybe is stupid because I don't know what the cross-linker is and why you're not saying what the cross-linker is. But what is the water absorption? Why aren't you mentioning the water absorption versus the others? Does it absorb more water, less water?
Yeah, for the first part, I mean, the cross-linker is not secret at all. We have the patents are published, and it's not secret molecule also because, as I said, it's used in other medical applications. So there are many published, a lot of literature on those molecules. It's just very technical, very chemistry. And for the water absorption, of course, we checked it, and the water absorption is quite low due to the low concentration of HA. So there are many ways to check for water absorption in the lab. But for consistently, we've seen the lowest or the equivalent to the lowest water absorption as compared to the current product. So there is no higher water absorption.
Interesting.
Just last question, really quick. I know we're out of time. Just the timing, your timing of your studies and your regulatory submission.
Yeah. So we are starting in Europe with a notified body, which I hope will get approval in Europe during 2024. And in US, we'll start also the studies, I hope fast, to get an approval in the beginning of 2026, I hope.
Great. Thank you very much.
Thank you.
Thank you.